Now let me show you how myosin interacts with actin filament and how it is able to move actin filament. I will also show you an animation in this module. So things will become hopefully very clear. So we are going to talk about this process in different steps. In the first stage, the head of myosin, it is lacking a nucleotide, is locked tightly onto actin filament. This tight locking of myosin head onto actin filament also causes rigorous mortis, which basically, when an animal or a person dies, their body becomes very stiff. The reason is normally cells have abundant ATP, and when ATP is bound to myosin, it does not bind to actin filaments very tightly, which is the next stage. ATP molecule binds myosin head in the back side of the myosin head, causing a conformational change of actin binding site. So when ATP is bound to myosin head, backside of the myosin head, it has caused release of this actin filament. This doesn't happen in people or animal once they die. So of course the body becomes very stiff. Next step is ATP hydrolysis. So we'll move to the next step. It is also called the Cork stage. Hydrolysis of ATP to ADP triggers a large conformational change, causing head displacement of 5 nanometers. First of all, let me point out cocked, the word cocked. Just like when people have to fire the gun, they first have to cock it, which means they pull the hammer back, may it be a revolver or a pistol. So basically, you are getting your gun ready to be fired. Also, this is the original position, the dotted line. This is the original position of myosin head. And this is the position it has assumed after it has hydrolyzed ATP into ADP. However, although ATP has been hydrolyzed, however, ATP and inorganic phosphate remain tightly bound to the head. The next stage is force generating stage. Myosin head now weakly binds a new site on actin filament, causing the release of phosphate ion, which results now in tight binding of actin myosin. In the next stage, head lacking a nucleotide, ADP has also left our myosin head. This head now is locked onto actin tightly, and it snaps back to the original position. Remember, in this stage, the actin had moved 5 nanometers in the forward direction. Now it is again in low energy configuration, if you want to call it. It snaps back, and now it has moved this actin filament 5 nanometers in this direction. So the plus end is here. This protein is moving towards the plus end. All things being relative, the motor protein is not moving. It is the actin filament which is moving along the head of this myosin head. This release triggers the power stroke, the force generating change in shape, during which this head has regained its original conformation. In this course of the power stroke, the ATP has been removed from this head. Now I'm going to show you an animation of this process, how myosin head attaches to actin filaments, and how they're able to move the actin filaments. So let's watch this animation. Here in the pink color, you have the myosin filament or the thick filament. These globular structures on the surface of thick filament are the myosin heads. The actin filaments are shown in blue. There are several other components in the system that we haven't talked about. We'll talk about them later, but here I just wanted to show you how myosin grabs onto actin filament. It converts the energy phosphate from ATP, high energy. It breaks the ATP, hydrolyzes into ADP and an organic phosphate, and it uses that energy to move the actin filament. So please notice. So here the calcium is released. I'll explain that later, but the muscle contracts in response to calcium release. it basically exposes certain sites on actin filament that can interact now with myosin. I'll tell you about that in more detail later, but right now we are concerned with interaction of myosin with the actin filament. So please notice how myosin heads will attach and they'll use ATP energy to slide these actin filaments. Let's see this process. So this process is going to repeat itself, and as you can see, the actin filaments are moving 5 nanometers per stroke. So now we are going to look at this more at a more higher magnification. The two heads are released. That we have two heads. I'll tell you again about these other components later on, but here you have two heads of myosin. So these are light chains that we talked about. These are alpha helical domains, coiled coil domains. We are going to remove one of the heads so we can see the process more clearly. Here's the head which is firmly attached to actin filament. This is going to just show you up close that the myosin head is closely attached to actin. This is what I said causes rigor mortis in absence of ATP. This is cross bridging between these two molecules. Now we are going to see. So here is the myosin head. It binds actin filament. It is pulling on actin filament. We will soon also see the ATP also. Now calcium has been reabsorbed, and myosin is no longer actin is no longer available to interact with myosin head again. But we are going to see this process again. How here's the calcium added in the system. This removes certain binding proteins from actin filament. Now actin filament is available to interact with myosin. Here the ATP is attached. This green structure is ATP. This little yellow dot is the phosphate, which is hydrolyzed and removed. ATP to ADP, and this molecule, this head of myosin, uses that energy to slide the actin filament. So this is basically the whole process. Again, hydrolysis of ATP causes a conformational change, resulting in sliding of this molecule, these two molecules against each other. I hope this animation was helpful in understanding how actin filaments move along the myosin filaments, which are called thick filaments.